Let's see. So far, we've learned about smallpox, yellow fever, and Ebola. Let me now formally introduce you to influenza virus, or more commonly known as flu. Flu is an enveloped virus whose genome is different. It's comprised of 8 separate negative strands of RNA, each coding for a protein or two. This unique feature has contributed much to the outbreak of massive multinational epidemics referred to as pandemics. The 1918 Spanish flu was such a pandemic, killing over 50 million people across 6 continents. More people died from this flu virus than soldiers killed in World War I. Flu is curiously a stomach disease whose natural reservoir is waterfowl. So, how do we catch the flu? In order to appreciate how this happens, we first need to understand some details of the replicative cycle of the virus. Let's take it step by step. Like all viruses, flu must first bind to its host cell. The flu spike protein, which binds to the receptor on the target cell, is called hemagglutinin and is designated H. In addition, flu has a second spike protein, the N spike, an enzyme called neuraminidase, that serves to cleave off the receptor protein when new viruses bud from infected cells, as we shall see later. The receptor for flu is a sugar called sialic acid, present on the host cell. Now, when the H spike binds to its receptor, the virus is able to enter the cell and release its 8 RNA segments into the cytoplasm. The viral RNAs then make their way into the cell nucleus where they can replicate. Remember, flu has a negative strand RNA. These must serve as templates to produce positive stranded RNA. The positive RNA can then be translated into viral proteins on cytoplasmic ribosomes. The same positive RNA can also be transcribed to produce thousands of copies of negative strand RNA genomes that will be packaged into new viruses that bud from the cell surface. When the new virus buds, its H spike may still be latched onto the cell surface sialic acid. In order to release the new viruses to the environment, it's the N spike that clips the receptor bound to the H spike and thus allows the virus to move on, seeking new targets to infect. As I mentioned, the natural reservoir of flu is waterfowl, where it generally infects their gut. Interestingly, there are different variations of H and N spikes, and so far, 17 H variants and 9 N variants have been identified in bird flu. These have been designated numerically in the order they were discovered. Thus, the first flu virus defined was designated H1N1. And over the years, all sorts of flu viruses have appeared in waterfowl population, such as H3N2, H5N2, and H7N9. So long as they stay within the waterfowl, flu doesn't present a serious problem. And in fact, the birds don't really get sick. But as you recall, Erica explained how viruses can spill over from an animal natural reservoir to humans. This process of evolving ecology of flu is what makes this virus so dynamic and consequently so deadly. When waterfowl migrate, they poop as they fly, spreading flu viruses along the way. They also visit ponds and streams near farms and villages where they contact domesticated poultry, infecting free-roaming chickens, ducks, and turkeys. When these farm birds get infected, they are inexperienced with flu, and so they develop serious respiratory disease. So long as H spike continues to bind to the form of sialic acid, unique to birds, the virus stays within the bird population. Here is where it gets interesting. Pigs are funny. They actually have 2 forms of sialic acid on the cells of their lungs. One is exactly the same as the bird receptor, while the other is a variation of sialic acid that is different, yet remarkably similar to that found in the lungs of humans. So think about it. When an infected hen sneezes on a neighboring pig, the pig gets flu. But now, the replicating virus has a chance to mutate and change, and through these changes, gains the ability to adapt and bind to the human form of sialic acid. Thus, the original bird-specific virus can infect the pig, and the new modified flu virus, mutated and adapted to the human form of the receptor, can emerge. So careful now. A sneeze from such an infected pig is contagious. The virus can now infect people, and an epidemic can proceed through person-to-person transmission. This process of adaptation through mutation of the bird's virus to the modified virus able to infect humans is referred to as genetic drift. Now, if a pig happens to be simultaneously infected with more than one type of flu virus, let's say H1N1 and H2N3, more complex mixing can occur. Remember, the genes of flu are separate, independent RNA segments, totally free to move about and reassort in any combination. The segments from different virus types can mix and match in the pig's lungs to produce novel combinations of viruses never seen before. This process of genetic shift produces viruses with dramatically new traits, often dangerous and lethal, for which humans have had no prior exposure or experience, and thus are particularly vulnerable and ill-prepared. It's the unique ability of flu to genetically drift and shift that constantly produces viruses that seriously challenge our immune system and yearly threaten us with the prospects of new and potentially deadly pandemic flu.